All right. Can everyone hear me if I talk like this? Are we good? All right. So I'm Dan Gates, and I'm from the University of Nebraska, and I'm going to talk a little about some of the dissertation work that I've been doing on some of the genomics of flower color transitions. And so we've had some great talks earlier on the importance selection associated with different flower color phenotypes. But flower color phenotypes, changes in flower color phenotypes, is also a frequently traversed path, which really can help or give us insight into the way that evolution operates because of the convergent nature of the trait and how frequently it occurs. But when we start to think about the trait itself, it's helpful to think also about the entire plant as well, outside of just the flower color, because the pigmentations themselves also can play, or the pigments themselves also can play important roles in other plant structures. So like in leaves, or in leaf tissue or stem tissue, oftentimes we see anthocyanin accumulation as a response to heat stress, and as well as an attractant for pollinators. And so this means that certain changes that may abolish the pigmentation pathway in the flowers and may possibly be adaptive, a new floral phenotype may also abolish the pathway in leaf tissues, leading to the loss of an important function. And so these sorts of changes are likely to be selected against, assuming that these functions are important in other tissues. And then we expect that if this is the case, evolution should operate in a predictable manner. We should see repeated fixations of traits that are modular in that they can change flower color, but maybe not influence anthocyanin accumulation abundance in other tissues. And so to really understand anthocyanins as a whole, and I'm going to talk about anthocyanins that tend to be the dominant form of floral pigmentation in flowers and in other tissues, we need to know how they're produced. And so this is the anthocyanin pigment pathway diagram here. And so what happens is you have an unpigmented upstream precursor, and on the left we have the boxes. Those are the structural genes, the enzymes that modulate the upstream precursor into the pigmented anthocyanins at the bottom. And then in the circles, we know some of the transcriptional activators, and then we also are learning more about transcriptional repressors as well. And so there's a lot of interesting components in the pathway, but I'd like to draw your attention to one thing that I think is interesting, and that is that at least 3 different transcription factors at different levels tend to be these MYB transcription factors, a certain specific type of MYB transcription factor. And there's probably a reason for that, in that the MYB transcription factor is one of the biggest plant transcription factor gene families, meaning that there's a lot of turnover, there's a lot of duplication, which can lead to subfunctionation, which can lead to elevated rates of evolution in these genes, and they also can lead to a high amount of tissue specificity as well. So it's been very fascinating watching other systems uncover the genetic basis of transitions from pigmented to unpigmented flowers, things like petunia, snapdragon, and mimulus, and columbines, phloxes, etc., because very repeatedly we see that there's a loss of function in this R2, R3 MYB activator, way more than you would expect just due to chance alone. And then the function of this, the expected function of this, is that when you lose this activation ability, that the pathway itself, where the activators act, gets turned down. You get white flowers through this change. And so we're interested in this in our plant study group, the subtribe Iochroma. It's a group of South American flowering plants in the Solanaceae, and you can see here, just looking at this phylogeny, that there's numerous different transitions from pigmented flowers to white flowers within the Iochroma. For what I'm going to talk about today, we're going to focus on a very recent transition from pigmented to a derived white flower phenotype, and that's in Iochroma loxensis. And so when we do the crosses, we see two important things. And the first thing is that the white allele appears to be mostly dominant, but it's intermediate enough that we can start to discriminate three phenotypes from these crosses. And then when we do back crosses, we see that it's likely a Mendelian large effect single locus that recreates the pigmented cyanium phenotype in the homozygous. You can see there's these intermediate heterozygotes, and then back crosses back to loxensis recreate the homozygous dominant or mostly dominant white or unpigmented phenotype. And so now that we know that we're looking for a single gene, we can start digging around in some genomics data and see if we can start to find genes that are segregating according to our expectations from these back-cross phenotype groups. And so that's what we've done. We used an RNA sequencing approach where we pooled these different back-cross phenotypes together. We sequenced them, and then, like I mentioned before, identified genes that were predicting phenotypes. And then we kind of get an extra bang for our buck when we do the RNA sequencing approach, because we can also quantify expression of those known pathway genes, because we can find those, the orthologs, and then also our candidate as well. We can start to quantify expression on that to understand more about the function of these types of genes. And so when we start to look for things that are predictive of phenotype, we find a single gene. It's an R3 MYB that ends up being our strongest candidate, and it predicts the phenotype almost perfectly. And so what's going on in this diagram, this is the contig of the pigmented parent cyanium, and then these little ticks here, these are known variants between cyanium and then the white flower parent loxensis. And so when we start to map different reads from these pools, we see no evidence of loxensis alleles, because we're calling no variants in this purple back cross. And then likewise in the heterozygote phenotype, we're seeing approximately even numbers of loxensis cyanium alleles, and then in the white back cross, almost complete loxensis alleles. And so it's an excellent candidate as far as segregation goes. But I also mentioned, I brought your attention to the MYBs earlier. There's some functional expectations that come from this too. So this doesn't just fit patterns of inheritance. It's a gene that is very similar to known repressors of pigmentation. Based upon studies in petunia, here's a control, and then here's transgenic. You can see it abolishes this red spot, these anthocyanins as well. So now, not only do we have a gene, but we have some expectations of how it should function. It should be a repressor based upon what we know. So we can then start to set up functional tests to test whether it's a repressor or not, whether it seems to have repressive abilities. And so that's what we've done as well. So since it's expected to be operating as a repressor, we should be able to overexpress it, and then it should abolish pigmentation. And so I made overexpression lines in Nicotiana. They're closely related to Iochroma within the Solanaceae. And sure enough, when we overexpress a putative repressor, we don't have nearly the pink flowers, the tobacco flowers that we see in the wild type. So this really helps us nail down, that we're really looking at a strong candidate here. But we want to learn more about it still, and so we can look at the expression levels, like I said, because we have this RNA sequencing data, and we can see here that functionally it seems to be operating by turning down the pathway. These bar charts, these are normalized expression on these pathway genes here, and you can see from F3' H down to ANS especially, down through F3'5' H, DFR, ANS, these downstream genes, they're almost completely unexpressed in the white flower the white flower back cross pools. So again, we're getting all of these lines of evidence that are leading us to understand, the gene that's involved and how it operates. but we can dig more into the structure of the gene itself and start to make some predictions about where did this come from. Has it always been serving this role, or is this a new thing for this type of gene? Like I mentioned before, there's a high amount of duplication, and these things constantly being subfunctionalized and neofunctionalized. And so we can look at the MYB gene structure. The activators we call R2, R3s because they have these two different domains, R2 domain, R3 domain. And the way that they work in the activators is that the R3 domain binds the transcription factor complex, and then the R2 domain binds the promoter DNA. Like I said, we know something about R3 repressors from petunia, things like that, and the ones that we have seen, they lost their R2 domain, so they only have the capacity to bind the transcription factor complex, not actually activate transcription, and that's what makes them repressors, because they bind that complex. Activators can't bind that complex, and then they reduce transcription in that way. And so that's sort of our null expectation, because this is the known function for these R3 repressors. And if this is the case, we know that these R3 repressors form a really distant, ancient clade within the angiosperm. So when we start to look for similar orthologs and things that might be related in closely related genomic sequences, we expect to see this deep divergence, this deep clade of R3s. But there are also a few documented reports of R3s being derived from a loss of R2 domain too. So it is possible that this could be a recent independent acquisition of this type of MYB repressor. And so I'm going to draw your attention to this alignment here. And what we can see is two things, that this first, it arose from a clade of R2, R3 genes. You can see this R2 domain is just abolished in this clade of MYB repressors here. And then the really interesting thing that we see is that the Arabidopsis MYB and the Petunia MYB, its close relatives that it's derived from, those are characterized repressors in their own right. And so this sets up a new hypothesis for the origin, is basically it came from a clade of repressors, it duplicated, lost its R2 domain, retained the repressive function. That's how it's working now. Turns out that's not right either, because it's also lost this active repressor domain at the end, and then independently acquired a new active repressing domain within the R3 region. So this is just a completely bizarre series of events. And so to put this sort of into context, what we're looking at is not an ortholog, it's not something that's similar to the expected repressor. Instead, it's duplicated and then undergone this elaborate series of domain loss and domain gains. And so what does this tell us about the repeatability of evolution? So there are a number of different ways that different mutations can lead to phenotypes. They can be the same gene, different genes with different functions. What we think, or what we're seeing here, is evidence that these are mutations in different genes than the R2, R3 activators that we usually see, but the function is retained, and that's repression of that pathway in the flowers. And this agrees with the idea that if you're repressing the pathway in the flowers, that pathway can still be active in other important tissues as well. And so, obviously the thing that we like to do in the future is start to walk out to the rest of the Iochromany here and start to see. So with that, I'll take my acknowledgments and I can take